Hi, this is Malinki. Welcome to my channel, Voice of Malinki. Hope you all are safe and doing well in this pandemic situation. So today I will deliver the third lecture of COVID-19 lecture series. That is how to treat coronavirus. Is this disease treatable at all? Is the disease curable? What are the current findings? And what is the best and most effective treatment? We will discuss everything today in a simple way. Hope you have gone through my first two lectures of COVID-19. If not, please check it soon. I am providing their links in the description box. And if you are new in my channel, please don't forget to subscribe my channel. And if you like my video, please do like, comment and share my video. Okay, so before proceeding to the action of different drugs, I will explain the action of virus in our cell, in our actually body cell. Very briefly I will explain. So otherwise it will be difficult to teach how the drugs act in our body. Now coronavirus enters our body through nose or mouth and travels through respiratory tract and enters lung. Now in lung, type 2 pneumocytes are there. Now these type 2 pneumocytes are the target of coronavirus. This is a type 2 pneumocyte. These cells have ACE2 receptors in their plasma membrane. And these ACE2 receptors are special type of receptors found only in these cells. Now the S protein or spike protein of virus has the high affinity for these ACE2 receptors. So, viral S protein will bind these ACE2 receptors. Once the binding occurs, the virus gets entered in the cell cytoplasm through the process of endocytosis. When virus gets endocytosed into these cells, there will be a fusion between endosome and lysosome. Now lysosomes will break down the lipid bilayer of the virus and viral RNA will come out of it. So this is the viral RNA. So the naked viral RNA, this is the naked viral RNA that is now in cytoplasm. Once the viral RNA is there in our cytoplasm, it will do two things. First of all, it will synthesize huge amounts of RNA. But how? Well, it will take the help of our host cell RNA-dependent RNA polymerases. These are some enzymes that generate RNA from RNA template. So, it will generate this RDRP, it will generate numbers of viral RNA from the RNA template itself in the cell. And one more thing the viral RNA will do. It binds our host cell ribosome. And binding of the ribosome and viral RNA will generate polyproteins. These polyproteins will be cleaved into smaller structural proteins like these by the enzyme protease. Now, these proteins include membrane protein, envelope protein, nucleocapsid protein, spike protein, etc. So, all structural proteins of the virus will be now made. They are ready. Right? Now, these proteins and the viral RNA that are only formed will combine to generate new viruses. And these new viruses are now formed and they cleave the host cell membrane and come out of the cell. Right? In this way, these type 2 pneumocytes are destroyed. So, this is the way how coronavirus is replicated in our lung cell. Now the first drug I will explain is remdesivir. Remdesivir actually blocks RDRP, this one, RNA dependent RNA polymerase. This remdesivir blocks this thing. Therefore, RNA synthesis is hampered, right? In that case, if viral RNA is not synthesized, new viral particles will not be generated. But who recommends against the use of remdesivir in COVID-19 patient, as there is currently no evidence that it improves survival and other outcomes in these patients. Although remdesivir is currently the only medication approved by the FDA to treat COVID-19. The approval was based on the findings that hospitalized patients who got remdesivir recovered faster. So, there is a controversy that whether this drug is effective or not. But currently, this drug is getting prescribed in some of the hospitals. Second is chloroquine or hydroxychloroquine. It acts in three ways. Number one, chloroquine is alkaline. It enters into the lysosome and increases its pH, thus decreases its function. When a lysosome can't work, fusion between an endosome and a lysosome will not take place. If this fusion doesn't work, viral RNA will not be released in the cytosol. Therefore, the production of viral proteins and RNA will not be possible. And in this way, multiplication of virus will be ceased. This is one mechanism. Next, a chloroquine is a zinc ionophore. That means it allows entry of zinc ions in the cell. Zinc in turn inhibits the action of RDRP or RNA-dependent RNA polymerase. Therefore, synthesis of viral RNA will be inhibited and viral multiplication is ceased again. The third way is chloroquine binds sialic acid found in plasma membrane of these cells. Now, the spike protein of coronavirus binds ACE2 receptor as well as sialic acid. If chloroquine binds this sialic acid, it blocks the binding between S protein and sialic acid. That is another way virus doesn't get entry in the cell. But based on these available medical data at this time, hydroxychloroquine is not effective for treating or preventing COVID-19. Though some small studies initially suggested that the medications may be helpful, many other large studies say they saw no benefit. The NIH and WHO have also stopped their own hydroxychloroquine studies after finding the drug had no clinical benefit. Use of this drug is causing some serious health issues or side effects like heart problems, blood and lymph system disorders, kidney injuries, liver problems, and failure. Next is ritonavir or lopinavir. This drug is a protease inhibitor. So it inhibits the protease. When protease gets inhibited, structural proteins of the virus are not getting produced from these polyproteins. Therefore, new viruses cannot be generated and viral replication is inhibited. So that's a cool thing. But lopinavir is a drug against HIV. This drug was regarded as potential agent in the fight against coronavirus. But unfortunately, it is now discovered that the concentration of this drug in the lungs of COVID-19 patients is not sufficient to fight the virus. Actually, when we use a drug, a particular concentration must be followed. If we cross the prescribed concentration and use it in a higher dose, it might be lethal for our body. There is a permitted dose also in case of ritonavir. But that much of concentration is not sufficient to kill coronavirus. If we use a higher dose, might be it will be helpful to destroy the virus, but it could harm us tremendously in the other way. That is the big problem. Next drug is tocilizumab or tocilizumab. So, if we remember from the first lecture that destruction of type 2 pneumocytes by these viruses release specific inflammatory mediators. Now, these inflammatory mediators will stimulate macrophages. These macrophages will now release specific cytokines like interleukin-1, interleukin-6, tissue necrosis factor alpha, etc. And these cytokines will enter the bloodstream and cause dilation and increase capillary permeability. As capillary permeability increases, plasma from the bloodstream will leak into the interstitial space. As it comes out, it will cause tissue swelling. That is scientifically termed as alveolar edema. Now, this excess fluid accumulation around the lung cell will generate a pressure that will impair gas exchange that may cause respiratory failure. And apart from plasma leakage, this capillary permeability will also allow these neutrophil influx into the lung. Neutrophil will release ROS or reactive oxygen species and protease in order to destroy the virus. But these ROS and proteases are not cell specific. So they will cause mass destruction of all alveolar cells including type 1 and type 2 pneumocytes. Type 1 pneumocytes have an integral role of gas exchange that will be damaged. Now type 1 and type 2 pneumocytes and other cells are all dead. Dead cells will generate cellular debris. That will lead to consolidation, or in a simple form you can say they will block the tissue. When tissue is blocked, again the gas exchange will not take place. That will cause low level of oxygen in blood or hypoxemia. When lung tissue is consolidated, you cannot breathe properly. You cannot take a long breath. This situation is named as dyspnea or shortness of breath. And tissue blockage will not allow to take a deep breath, so you will cough. Here I have already mentioned that dilation occurs due to these cytokines, right? That means blood pressure will drop. As the these blood vessels are now wider, so definitely blood pressure will drop. This will cause hypotension. Hypotension will cause decreased perfusion that causes multi-system organ failure or MSOF. If MSOF or multi-system organ failure occurs, we cannot survive anymore. So one thing we can understand here that cytokine is the main culprit here. The cytokines, they are the main culprit. It causes these all downstream effects I have just mentioned. Now there is an antibody against interleukin 6, that is IL-6. So that is the main cytokine in the mechanism. So this antibody is this. That is tocilizumab or tocilizumab. It is manufactured in Roche. This drug is little bit effective and recommended by some doctors. Next is corticosteroids. This is one more drug like tocilizumab that reduces inflammation and somehow protects us. WHO is strongly recommending this drug for the treatment of patients with severe and critical COVID-19. Now the last thing is convalescent plasma therapy. This therapy uses blood from people who have recovered from COVID-19 to help others recover. The FDA is allowing its use during the pandemic because there is no highly effective treatment for COVID virus, right? Suppose this is the person who is infected by coronavirus. Now he will generate some antibodies against the coronavirus. The antibodies will be mainly IgG types. So he will make these IgG antibodies in his body. But it also takes some time to produce IgG in our body. Now when this person is recovered, his blood will have ample amounts of this IgG. So now this person can serve as a donor. So we can take his blood. The blood is now processed to remove blood cells, leaving behind the liquid part of it or plasma and IgG antibodies. Now these antibodies or these things can be given to a people with COVID-19. He is now a recipient. It will boost his ability to fight the virus because he is getting some ready-made antibodies now that will bind the viral antigen directly. But AIMS chief Dr. Randeep Guleria said that antiviral, remdesivir and plasma therapy have a limited role to play in the treatment of COVID-19 infected patients and are not of much use. So, drug is not working effectively in this disease. Although we are getting some positive results in the laboratory in a small scale, but practically those are not very much effective. So what to do? How will we get recovered if infected? Will we all die? No. The only hope is our immunity. Even we take vaccine also, we may get this infection, but it is assumed that the severity will get reduced in that case. That is a different story. We will discuss about that in the next video. But currently, the ultimate weapon is our immune system. That is defending tremendously for us against the fatal virus. Please take good food, rest well, sleep for at least 7 hours a day, lead a good and healthy lifestyle. These all will help to boost your immunity. And the last message is to stop stressing and panicking. Stress reduces our immunity several times. That is scientifically proven. We will discuss the direct relationship between stress and immunity one day. Keep faith on your immunity. If you're a student, your exam is postponed or cancelled, if you lost your job, if you're searching for a job but not getting, don't be stressed. Don't be depressed. It will just welcome Corona. Try to avoid crowd, be in home, stay happy, stay safe. Stay with me for the next exciting video. Bye bye.